Hello, my name is Steven Zeldin. I am a senior in Columbia College, majoring in neuroscience and behavior on the pre-medical track. Working at a lab at the New York State Psychiatric Institute, the Baldrini Lab with Dr. Maura Baldrini, I have been researching the proteins linked with the aging of the human brain, and specifically with the aging of the human hippocampus, a region that has been implicated with long-term memory. Effectively, whereas aging, of course, leads to various deleterious effects, including changes in neuron function, neuron connectivity, and the macroscopic structure of the brain, and of course, use its deterioration, which we have not, for the entirety of our species' history, been able to remedy. With the advent of new tools to study the proteins made in different parts of the brain, it's now, for effectively the first time in human history, feasible to see which proteins change in concentration in the brain as the human body ages. Thus, over the course of my project, I sought to determine which proteins change in quantity in the brain as patients aged. Using human models with people who donated their brains upon death to the Psychiatric Institute, I conducted a series of studies using mass spectrometry to determine which specific proteins were present in the hippocampus for each patient who was a different age. By comparing the quantities of these proteins across these different patients who had different ages, I was able to determine which proteins increased or decreased in general as people aged. Overall, I found that some extremely important protein groups that change in quantity as patients age include those related to lysosomes, those related to the cytoskeleton, and perhaps most important of all, those related to the mitochondria. This could show that those three key areas are very important in aging, where dysregulation of proteins related to those three functions could lead to many of the effects we associate with aging today. Thus, with this study and others in the future, we've opened the door to understanding aging at a never before reached scale. Thank you very much for your time.